Okay. So good morning or good afternoon, wherever you are. It's really nice to see you here. Quick intro for those I haven't met before. I'm Louise Perrault. I've spent the last 20 years working at the crossroads of evidence and access, helping pharma, biotech, and medtech companies figure out how to tell the story of their data, what story to tell with the evidence in a way that actually reaches the people who make decisions. People like regulators, HTA bodies, payers, and even sometimes clinicians. So here's a quick overview of what we'll cover during the session. I'll start with rare disease trials. We'll walk through the case study design, review the key results, and explore how a Bayesian approach deepens our understanding of a trial initially dismissed as futile. We'll wrap up with some conclusions and takeaway. So let's dig into it. Let me set the scene for the challenge we are dealing with. Most of the statistical tools we use in clinical research were built for big studies. Hundreds, sometimes even thousands of patients, stable patterns, predictable results. In that world, the math works perfectly. In rare disease, though, it is a completely different world. Sometimes you have 30 patients, sometimes you even have fewer patients. The patients vary a lot from one to the next. There's lots of heterogeneity. And more often than not, you do not have much historical to go on. And the disease can often be aggressive. So in that environment, a small difference in the data can swing the results dramatically, and the standard tools, which rely on having enough patients to be convinced, can easily miss the real signal. So I want to spend a couple of minutes on what is a p-value, and specifically on what is not a p-value. Most of you have seen p-values in trial results your whole career, but I find that even experienced people who work with data day in and day out, they have a version of its interpretation in their head, and that is their unique way of interpreting a p-value. Most of the time, it is not statistically significant. It fits over the p-value of 0.05. What does that really mean? P-value over 0.05. So the p-value tells you how surprised you should be by your results if the drug doesn't work. So under the null hypothesis, the p-value tells you how surprised you should be about your result. What the p-value does not tell you is the probability that the drug works. These are two different concepts. So a p-value above 0.05 is not statistically significant. However, it does not mean the drug does not work. It means we do not have enough evidence to be sure within this framework of the standard method, the frequentist method, that the drug, that there's not enough evidence. And in a rare disease, where your trial might have 30 patients or less, you can have a result that looks genuinely promising and still get a p-value of above 0.05. And this is not because the drug failed, because it's mainly because the trial was too small for the test to feel confident about the evidence. So that's the central point at the heart of today's case. And it is what pushed us to ask different questions. The case involves a drug called ARI. It was tested in patients with a rare inherited heart condition caused by a mutation in one specific gene. I won't get deep into the biology because this is not the point of this presentation, but here's what you need to know about the disease. It tends to hit people young. It causes progressive heart failure. Patients are at high risk of sudden cardiac death. Many end up needing a defibrillator or even a heart transplant. So this is pretty serious. The unmet need is real. These patients do not have any other good options, and their disease deteriorates quickly. So there had been a smaller study, a phase 2 study on ARI in 12 patients where the drug looked really promising. Patients improved meaningfully on several measures. That was enough to move forward to a full phase 3 study. The phase 3 trial was properly designed. It was randomized, blinded, placebo-controlled. It had a planned interim analysis, and at that point, the committee looked at the data and concluded futility. The result was not statistically significant. The trial was stopped. So here are the numbers on the slide. Risk reduction estimate after the phase 3 study was 57%. The range of uncertainty was very wide, and the p-value was 0.23, which is well above 0.05. But think about that for a second. The data were pointing to a 57% lower risk of getting worse or dying. That is not a small signal, but the result was so imprecise, the range of uncertainty so wide, that the test could not rule out the possibility that it was just chance. So the official conclusion was not significant, and the program was stopped. But imprecise is not the same as wrong, and uncertain is not the same as absent. So we asked, what else could we have learned from these data before we close this case and walk away? Here's where we dig deeper by using a different approach, which is the Bayesian approach that combines prior knowledge to the data you collected and obtain an updated estimate which gives you a very interesting piece of information. We said, okay, we already knew something about this drug before the phase 3 trial started. We had the earlier study, the phase 2 study with 12 patients. We had the biology also. And in the phase 2 study, there was a signal pointing in a particular direction, obviously the right direction. So instead of ignoring all of that data, we asked, what happens if we formally bring it into the analysis? So the Bayesian analysis does that exactly. So in our approach, we started with what the earlier 12 patient study told us, where about 7 out of 12 patients responded to the drug. That became our starting point for the analysis. Then we took the phase 3 data, the 57% lower risk with its very wide range, and we combined the two. What we get when you combine the two studies together is an updated estimate. And because you're pooling two sources of information, the combined picture is more stable than either one alone. The range of uncertainty narrows. It doesn't go to zero, of course. There's still uncertainty, but you have more to work with. our updated estimate came out to roughly a 46% lower risk. It still had a wide range. It was still crossing the no effect, the one line, but the overall picture was more informative than either the phase 1 or the phase 2 alone, not combined. Here's the part I find most useful from Bayesian analysis, especially for people who need to communicate evidence to decision makers. And you can see, in this example, there was a 94% chance that the drug reduces the risk for patients. So that's just reducing any risk. And you can go even further and say there was an 84% chance the benefit was not just marginal, but clinically meaningful. The kind of reduction that would actually matter in someone's life. Now compare that to p-value equals 0.023, 0.23, not statistically significant. Which of those is easier to work with in a meeting with a payer, in a conversation with a regulator, in a dossier when you need to make the case for a drug in a disease with 30 patients in the world? The underlying evidence is exactly the same. What changes is the language, and the language changes maybe what decisions are possible. Depending on your role, you will be able to communicate differently. I just want to make this concrete example from where you sit. Let's say, as an example, you are a person who works with HTA bodies and payers. Your classic discussion is, do we have enough evidence to justify the cost? Well, with a Bayesian framework, we can discuss the chance of a meaningful benefit. So a fair question to ask at this point is, well, you chose what to bring in for the earlier study. What if you had assumed something different? Would the answer change? That is exactly the right thing to ask at that point, and it's exactly what we tested. We ran the analysis 4 times, each time with a different starting point. One starting from what the earlier study actually told us, the phase 2 study, one's with a more optimistic starting point. So we assumed that the drug would work quite well. One's with a cautious starting point, assuming it might not do much, and one's with no starting point at all, as if we had never heard of this drug before we started the phase 3, which is, in my view, quite impossible, because this is not how drug development works. Usually, you run a phase 2. If the results of your phase 2 are totally negative, then you don't move on to phase 3. So you know some information before you start your phase 3. But we ran this analysis for the skeptics in the room. So even when we walked in with 0 prior expectation, no reason to believe the drug worked at all, the phase 3 data alone still gave us a 76% chance of some benefit. That is a signal that persisted no matter how skeptical we were going in. And this is an important point about how this approach compares to the standard one, to the frequentist approach. The standard approach also makes assumptions. Don't be fooled by the frequentist approach. It also includes assumptions. It includes assumptions about how big the effect is expected to be, the dropout rate, baseline event rate, what counts as a meaningful result. The difference is that in the standard approach, all these assumptions are baked into the sample size calculation and in the trial design. And these assumptions are not revisited most of the time after the trial starts. In a Bayesian analysis, the assumptions are on the table, and we can even change them, modify them as we wish to look at the robustness of the results. To me, that transparency is a feature, not a limitation of the Bayesian approach. It is what lets you have an honest conversation about what the data cannot support. So I want to zoom out for a moment before we close. The paper points to something important about this specific case. Looking back at the phase 3 trial, there are reasons to think the futility call may have been less to do with the drug and more to do with the trial design itself. Some patients enrolled had a version of the gene mutation where it was not clear they would be used, had not been specifically validated in that patient group. So with that kind of variation in the room, even a drug that works in most patients can look weak across this whole group of patients. And here's an interesting data point. A follow-on study enrolled only patients that clinicians believe were most likely to benefit. Over nearly 3 years, those patients maintained their ability to exercise. That's a meaningful finding, and it suggests that when you find the right patients, the signal gets clearer. So a trial stop for futility might be telling us the drug did not work, or it might be telling us we were asking the wrong patients, measuring the wrong things, or using a framework that could not hear the signal. Those are very different issues. I'm not saying every stop trial should be reopened. I'm saying we owe it to the patients, especially patients with rare diseases who have no other options or very little options, to make sure we are learning everything we can from every study that gets run. These trials are very expensive, as you know. They take years, and real people participated in them. The least we can do is make sure we read the results carefully. I want to close with something practical. The next time you are sitting in a development meeting, developing value messages, reviewing a dossier, or talking with a payer about a trial that did not quite hit significance, I want you to have one extra question in your toolkit. Is the result telling us the drug failed, or is it telling us we did not have enough patients for the test to hear the signal clearly? Because those are different situations, and they may deserve different responses. The approach I talked about today does not replace the standard way of analyzing trial. It sits alongside it. It is what you reach for when the classic analysis gives you a close call and you need to understand what the data is really saying. The shift is not complicated. It's really just this. In rare disease, where every patient matters and every trial is hard to run, that shift in question can change what gets developed, what gets approved, and what gets to patients more importantly. Thank you. I'm really looking forward to your questions.
Great. Thank you so much, Louise. There are some questions that already came in. One of the first ones relates to what you were just talking about in terms of trial design problems and asks, how does Bayesian analysis or reanalysis address those issues, such as mixed patient population or unvalidated endpoints?
Yes, that's a very good question. Thank you. Can you still see my screen?
No, we can't see your screen now.
Okay. Okay. I thought I had stopped sharing. So, yeah, so you said how, I'm sorry, you were talking about mixed patient population and unvalidated endpoints. Is that what you were talking about, Nicole?
That's right. And the question
Oh,
asks,
yeah.
how does, yeah, thank you.
Okay, yeah. Well, I have to say, it really doesn't fix anything. And that's an important caveat. And thank you for the question, because this is very important. Bayesian analysis can't fix a study that was set up in a messy way. It has to work with the data that was collected. So if the trial mixes very different kinds of patients, like we saw in our example, or measures an outcome that's hard to pin down, or any issues like this, the results will be noisy no matter what statistic we use. The Bayesian method can still help us by being upfront about the uncertainty and showing how much the data lean towards benefits versus no benefit, but the design criticism is still a separate issue. So the trial might look like futile because of how it was built, not because the drug truly doesn't work. And Bayesian reanalysis can support some of that, but it cannot rewrite the trial after the fact. I hope this answers the question.
Thank you. So the next question that came in is more technical. What is the difference between confidence interval and credible interval?
Oh, that's a very good question. And thank you for asking that question. In fact, these are two different ways to talk about a range around the study results. A confidence interval, like the 95 confidence interval, is a reliability statement about the method. If we could run, I mean, the credible interval says if we could run the same kind of study over and over and over again, and each time we would have a confidence interval, then about 95 out of 100 of those confidence intervals would cover the real truth. That's what a confidence interval is about. But for the one study you actually ran, it doesn't really mean there's a 95% chance the truth is inside the interval. This is not what it means. It's kind of like the intuitive way we think of a confidence interval, but that's not what a confidence interval. The 95% credible intervals that come from Bayesian analysis, this is closer to what we naturally mean. Using the data from your study and whatever starting assumptions you choose, which is the prior, which was the phase 2 study in our analysis, credible interval says there is a 95% chance the true effect is somewhere between the two numbers. So it's a direct probability statement about the thing you care about, which is the true effect. It's a little bit difficult to have the feedback to see if I answered the questions properly, but I hope I did. I'm ready for the next question.
Thanks so much. Okay, this one is a bit technical, so I hope I get it right. It's a bit of a scenario. So in a sensitivity analysis, even the non-informative prior gave an 88% probability of benefit. So I think this is a scenario that they're describing.
Mm-hmm.
And the question is, why doesn't a non-informative prior just reproduce the frequentist p-value? Very technical.
Yeah. So what the question is, just for people, is that if you have no prior at all, which doesn't really fit with a Bayesian because you need to have a prior. But basically what that means is that we assume at the beginning for the prior that we know nothing about the drug. And then, we look at the data from the phase 3 study and combine both of them. So because the posterior and the p-value answer two different questions. So the p-value asks, if the drug does nothing, how often would I see results at least this extreme? So when you get a p-value of, let's say, 0.23, like in our example, this is how often you would see results that extreme. The posterior probability answers a different question in a Bayesian analysis. It says, given this data, what is the probability the hazard ratio is below 1, meaning it's effective? So these are two distinct, they're distinct mathematically, basically. With a vague prior, the posterior is almost entirely driven by the likelihood, and the likelihood was the phase 3 in our example. But it still expresses the results as a probability of direction rather than a pass or fail threshold. So there are two different types of analyses.
Thank
The
you,
next
Louise.
question, please.
Well, it looks like we're at time, so
we could. Okay.
Yeah, we could follow up with answers individually to the remaining questions.
Absolutely. It will be a pleasure.
Thank you so much for your time and the wonderful presentation. It was truly enjoyable. And we look forward to responding to the remaining questions individually.
Yes, and I look forward for the next webinar.